Hello children, welcome to Vivid Botany. We are looking another question of 2020. We have been given here four enzymes. These enzymes are not the conventional or traditional enzymes that catalyze the metabolism of the plant like photosynthesis, respiration. These enzymes should be perfect with molecular base of inheritance as well as the biotechnology. Let me discuss the enzymes one after the other. The first enzyme is given here as the ligases. So ligase are those, actually ligase are nothing but the DNA ligases, otherwise called as they are molecular glue. The synonym of that is molecular glue. What do? They join the two DNA molecules. For example, I'll give you, we have discussed one question that is EcoR1. EcoR1 is restriction endonuclease which will cut the DNA in between G and A. GAA, TTC 523, GAA. Okay, let me write bigger. GAA, TTC from 5 to 3 and GAA, TTC, this is from 5 to 3. So EcoR1 will cut in between exactly G and A from 5 to 3 like this. So the cutting will be like this. This is one broken fragment of DNA and this another broken fragment of DNA. Remember, the restriction endonuclease will cut ester bond. Ester bond lies in between sugar and phosphate, I have told you earlier. So these ester bonds are cut and they are not cut at same point. They are called as staggered cuttings. As a result, the cohesive sites are formed here. Two complementary sites or sticky ends are formed here. So these are the sticky ends that are formed, and the sticky ends readily unite with each other when they are coupled with the DNA ligase. So here we find the G, C, and here we find the T, T, A, T, T, and here we find that C and G. So these bonds, these two fragments of DNA will readily unite by DNA ligase. So do remember that DNA ligase, which is called as molecular glue, will unite or join the two strands of DNA. The two strands of DNA that are complementary to each other, or you say the sticky ends of two DNA. Two sticky ends of DNA are readily joined here. In fact, it forms here, remember, it forms here the how many hydrogen bonds here? 2, 2, 2, 2. That is 8 hydrogen bonds it will form, and 2 ester bonds. 2 ester bonds and 8 hydrogen bonds are formed when the two cohesive sites of EcoR1 are joined by DNA ligase. And what does it cut exactly? It cuts only 2 ester bonds. Hydrogen bonds are cut they themselves. But when it joins, 2 ester bonds as well as 8 hydrogen bonds are joined together to attach these two fragments. So that is the function of DNA ligase or molecular glue. Let us see next. The second one is polymerase. So what is the polymerase? Polymerase is that which causes polymerization of nucleotides. It catalyzes all their enzymes. You can call it as catalyses. Catalyzes polymerization of nucleotides. Nucleotide, you must be knowing nucleotide, that is sugar plus phosphate plus nitrogen base is called as nucleotide. In fact, they cause polymerization of nucleoside triphosphate. Nucleoside triphosphate. What are nucleoside triphosphate? Let us see, this is sugar. Sugar is attached to the nitrogen base. May it be like adenine, guanine, cytosine, thymine, uracil. So this is nucleoside attached to 3 phosphates. 1, 2 and 3. So this is called as nucleoside triphosphate. So polymerase will cause polymerization of nucleoside triphosphate. These polymerases are two kinds, that includes here DNA polymerase, another is RNA polymerase. What is the difference in between DNA polymerase and RNA polymerase? DNA polymerase will cause polymerization of deoxyribonucleoside triphosphates. Yes, it causes polymerization of deoxyribonucleoside triphosphate. Nucleoside triphosphates. I write here tri-P. They include here adenine, guanine, cytosine, thymine. And RNA polymerase will cause polymerization of ribose nucleoside triphosphate. They include here adenine, guanine, cytosine. There is no thymine but uracil. So simply speaking, DNA polymerase is helpful in formation of DNA. RNA polymerase is helpful in formation of RNA. So we know that RNA polymerase will form RNA in the transcription. Other name of RNA polymerase also called as transcriptase. It's called transcriptase. Do remember, the transcription, it takes place in nucleus of eukaryotes. In case of prokaryotes, it takes place in the cytoplasm itself, because there is no compartmentalization of nucleus and cytoplasm in case of prokaryotes, as there is no nuclear envelope in case of the prokaryotes. So that is polymerase causes polymerization of the nucleosides, or we can say it causes the formation of DNA and the formation of RNA. Third one, nucleases. Nucleases are the enzymes which cause digestion, which catalyze digestion of nucleic acids, namely the nucleic acids, namely DNA and RNA. So for DNA we call it as deoxyribonuclease. For RNA we call it as ribonuclease. And this is deoxyribonuclease. So ribonuclease, deoxyribonuclease. So again, these nucleases are two types, like we call exonucleases and endonucleases. Let me tell you what are exonucleases. This question is been asked in 2021 also. One should be vigilant and try to understand exactly what do they mean. So exonucleases are those which cut the end of nucleic acid, They are called as exonucleases. And endonucleases are those which cut specific site, यानी कि palindromes. We know palindromes, I wrote here GATTC. Like that, every resistor endonuclease has got its specific palindrome. At that palindrome only the resistor endonuclease will cut. Such are called as endonucleases. So they are called as palindromes. What are palindromes? Which are same when you read in opposite direction. Like we have Malayalam, we have race car, like that. I have told you some examples in our previous question, NEET 2021. So these are different kinds of enzymes are given here. You should have a thorough comprehension to understand the question. Now he is asking what is the correct pair among the following. So ligases, they join the two DNA molecules. It is correct. Option 1 itself is very much correct. Remaining polymerases, they break DNA into fragments. No. Which will break DNA into fragments? Nucleases. Right. Or else we can call it a deoxyribonucleases. They cause breakdown of DNA into fragments. Nucleases, they cause digestion of DNA. They cause digestion of DNA or RNA. They are called as ribonuclease, deoxyribonuclease, exonucleases, which cut specific sites. This is wrong. They won't cut specific sites. They cut the ends of DNA. They are called as exonucleases. So I think my explanation is very clear to you. It is very much helpful to you. So we will be trying to elaborate, expand and detail discuss each and every question that's been asked in NEET 2020, right? So this part of question is taken from molecular biology as well as biotechnology. It is very good question, very shandar question being given from the modern biology, application part of biology. So thank you for listening to me. Please drop your opinions also. Please comment on that. You want any change, you want any other matters to be explained, I am ready to explain you. We will come together, we will meet together and I will be explaining you one question after another. Thank you very much. So please subscribe my channel Vivid Botany. Thank you.